So thank you very much. So I want to talk a little bit about, I want to talk a little bit about the work that I do. So it's not so much a technical lecture, but I want to give an overview of the type of thing that I do. In truth, the work that resulted in the Ricci Prize was very much tangential for me. This was sort of a one-off. It was a happy sort of coincidence that we had an interesting problem from Kyong and some interesting students, but I do want to try to fit it in at least to some type of broader perspective. So the title of the talk is Hidden in Plain Sight, and it really encompasses the two types of problems that I work on. The first one is finding things that may or may not be, or finding things in images that may be somewhat difficult to discern because their contrast isn't good or there's clutter. And the other one, this is actually Morse code for sight. So the idea here is that we're trying to decode information. So I want to give an overview. So this is really a talk about these two problems, these two classes of problems, and some applications or some places in which I've been lucky enough to work. So the first problem is one of image segmentation. So let's step through a typical image segmentation problem. If you have a picture of someone, say Steve Ricci, right? You may want to do, this is all I could find on the web. I process images, but I don't take very many of them. So the idea here is you may want to do something to this image. For example, I purchased a new Samsung Galaxy S3 the other week, and I found that not only does the camera do red eye correction, but it finds smiles. So I pointed the camera at my daughter, one of my daughters, and I pressed the button, and it waited, and then she smiled, and it took the picture. Okay, so this is a typical image processing problem. So the way that I formulate these problems, or I look at these problems, is that you have this, someplace in the real world we have Steve, and we collect some information about him through a sensor, in this case a camera, and here's my somewhat lame attempt at a pixelated representation of Steve. I am no artist. And the idea behind the problem is that we're going to do something to this image. So in truth, on a camera, this computer is actually embedded in the device itself, but we want to find something. And I was thinking of all the different things that you could find, and here's the simplest one would be that we want to find his face, right? If you wanted to do facial recognition. There are other things that you could find. If it were me, I would be looking for the books in his office, because while I like him very much, I always like to find people's offices. It's the month of being chair. I've lost most of my hair, so maybe I'm interested in his due. So you could do all sorts of things. And this is one class of problems. So you're given you take a picture, you have a sensor that collects tons and tons of data. And I think of the cameras or microscopy. You could imagine video surveillance. So the general problem here is image processing. And these problems in image processing, are characterized by an abundance of data. So if you look at these types of sensors, these types of instruments, cameras, microscopes, video, you're talking about gigatera, or for some real problems, petabytes of data. And the issue there, the primary challenge here, is in fishing out what you're interested in. What is it that's interesting in these pictures? What happens if what's interesting in this data? What's different? Normality is generally easy to find but uninteresting, but what's different sometimes? So in this particular problem, which we'll look at a little more later, we're interested in cell. This is electron microscopy of a mouse brain, I believe. It's some kind of brain. We're interested in cell boundaries. In this problem, this is a 3D. I think it's either CT or MR of the vasculature in the brain. And here the issue was, can we find in all of this mess aneurysms, and then can we say something about whether or not those aneurysms are going to blow, right? You want what's the likelihood of rupture? This is a problem from Homeland Security. This is an X-ray backscatter, type of data you get when you go through an airport. There the issue is finding, with very low false alarms, threat objects, say. And lungs aren't threats, yet they do represent some kind of perturbation, so how can you be robust? And this is an image that we've been playing, a type of image we've been playing around with Professor Georgia Cootie. Here the stripy things here, the texture is collagen in a silk composite, all things silk here. And we're interested in sort of quantifying or saying something about the structure of the collagen. So the challenges for these problems and other problems are really how can you extract interesting information in finite time with finite resources and with little or no human interaction. So how can we get a computer to sort of sort through this mess and come up with things that are salient or interesting? That's the in some sense, one of the fundamental image processing problems. A separate set of problems is more image formation. And this is, again, we're going to start with Steve, and now we're interested in really understanding what makes Steve tick, right? What's in his head that makes him so successful and makes him so generous to Tufts? And if you want to figure that out, well, I would go to a CT machine, and we'll stick Steve in the CT machine, and we'll collect data. This time, the data that's collected look nothing like Steve's head, right? This doesn't look like Steve's picture. This is, in a sense, well, this is the raw data collected by a CT machine or a representation of it. This doesn't look like anything that you or I would be able to interpret, and it really shouldn't. This is called the radon transform of something, but still we want to process it. Somehow, the people who built this system or similar systems have found a way of taking these data and reproducing a cross-section of Steve's head. Okay, and the idea again is that there's a computer or there's some computational device, and what pops out is this. So this is what Steve's thinking about. I think the reason why he's so generous and why he's so enthusiastic about Tufts is he's always thinking happy thoughts. When in my 3 years as Associate Dean of Research, he was always one of the happiest people that I ever, and the most upbeat people that I ever got to meet when I was sitting through the at that time, Board of Overseers meetings. And I attribute this to, and I think that this little experiment just shows one of the reasons why, that it's really deep inside that we have this kind of enthusiasm. These problems, unlike, say, the image processing problems, are in some sense opposite. So here I've provided some indications. So there's of different types of systems. So for example, CT or MRI is certainly one that most of us would know about. But there are a lot of interesting applications for these types of, a lot of interesting problems where this comes up in, for example, subsurface sensing. This is, I think, probably a ground penetrating radar. The data it collects looks like this, and the question is there a mine under the ground? And you can definitely see that there's something under the ground here. This is the ground surface. Is it a mine? Is it a rock? What can we tell? Here's an example, a different type of tomography problem. I think of these as tomography. This is electrical resistance tomography, where somebody's bore holes into the earth. And the idea here is that I'm going to put little current sources along here and little voltage sources here, and I'm going to force electrical current into the earth. And I'm going to listen for the voltages at other boreholes. I'm going to change where the current is, I'm going to change where the voltage is, and then I want to build a map of the electrical conductivity in the region sort of that's being interrogated. So this is kind of like tomography, but it's a really warped kind of tomography. This is a very hard problem, but the idea is that things that are in the earth, oil, petrochemicals, contaminant plumes, have some contrast in electrical properties between that relative to the background. So maybe we can tease out of these very strange data, these very limited data, the things that we're interested in. And here's an example, so we'll talk a little bit more about this. This is a project that I've worked on with Linda and her group and the civil engineers on using flow and transport. So this is far, far from electrical engineering. This is a problem in hydrology where some of these ideas of image formation or sort of media characterization can also be applied. So the problems here, all of these problems, are different. I mean, in a way they're the same, but in a way they're different. So the physics of the sensing modality become more important here. Whereas a camera gives you pixels, which are more or less proportional to what we all see, these things give you data sets that are, whose visual inspection tells you very little about about what's going on. The data are also often constrained. So here in medical CT, the reason medical CT works so well and MRI is you can take that person, you can put them right in, and you get a view the whole way around. Same thing is true for luggage screening. And that's really helpful. In fact, the Nobel Prize for CT went to, I think Tufts' only Nobel Prize was for Cormack, who was a physicist in the 70s, I believe. And he was the one, he and I think Hounsfeld shared the Nobel Prize. Their contribution was figuring out how to take that data and produce these exquisite images. And the reason the images from CT and MRI are in fact so useful and so exquisite is that you collect just a ton of data. But most of these interesting tomography problems, you're very constrained with data. Here, I can't drill boreholes everywhere. I can't encircle the region. Here, we definitely can't encircle the region, and in this case, I'm only allowed to take radargrams from the surface, in sort of a backscatter geometry. So we really have problems with very little data. And the issues there are how can we stably recover something interesting about the region that we're interested in imaging. So, and stability here is a big issue. You sort of have, when one tries to invert for pixels or recover sort of a dense pixelated version here, I really don't have enough data to support that reconstruction. In a linear case, and it's not a linear problem, if you're a linear algebraist, you're looking at a matrix that's sort of short and fat. And anyone who's dealt with short, fat matrices knows that there's a null space. And essentially, it's the null space. It's all in a very precise mathematical sense, there are a lot of things here, a lot of ways of distributing these pixels that are invisible to the data, that give no response to the data. So we come up, a lot of the work in my group is coming up with ways of sort of sorting through all those possibilities and hopefully picking one that makes at least a little bit of sense. Okay, so my research area, and the reason I was recruited from, or reason I allowed myself to be recruited from Northeastern, is that I really sort of sit between sensors and information. So I like building these algorithms that turn data and sensor physics into something somebody actually wants to use. And for me, Tufts has been like my candy store. If you had to talk about, if you're a kid in a candy store, I could have thought of nothing better than coming here 5, 6 years ago, because I just saw such a plethora of opportunity for myself. This tends to be, the stuff that I like doing tends to be reasonably interdisciplinary, right? I work with folks in hydrology, we'll see. So it tends to be reasonably interdisciplinary. So there were a lot of opportunities here to work. Tufts has a medical school. It's been great. And really, it's the ability of the graduate and the undergraduate students that lets this, it's sort of these abilities of these students that kind of lets this flourish. So the types of problems that we've been working on actually are more or less, this is more or less current. I think, one of my students just defended today, look like this. So we do about half the work in my group is related to these tomographic problems and half to image processing. So I've listed here, and I've listed the PIs that I work with. So we do work in diffuse optical tomography. This is using near-infrared light for breast cancer detection. That's a nice joint project with Sergio and Misha Kilmer. What I want to talk about a little more in a minute is work in hydrology with Linda, Kurt Pinnell, and Andrew Ramsberg. We also do some work on X-ray tomography for baggage screening. On the image processing side, there's sort of the fat-finding problem that Kyung has wrapped me up in, and we'll talk about. But we also do work with Irene. We do some interesting work on neuron tracking with some folks at Harvard, and as well as some folks at Brigham and Women's. And then you could kind of see us here with angiography. So this is with a neurosurgeon at Tufts. We've done a lot of work in looking for quantifying osteoarthritis from knee MRIs with Tim McElinden from Tufts Medical Center, and a lot of work on processing X-ray backscatter data. So this is the airport security data to try to find anomalies. And it's always fun to work with people from industry, and we have a nice relationship with American Science and Engineering. So I've loved it here at Tufts. I mean, I think it's just great, and I've had the best time and continue to have the best time. So I wanted to just touch on 2 of these problems. So this one I just love talking about. I love talking about all the problems, but this one, I had a student that was a really good artist, so it makes it easy to talk about this problem. And see, she has the prettiest results and some really cool technology. Then I want to talk about this problem here, because this is the one that really led to the Ricci Prize. Okay. So one problem that we worked on that I'm particularly proud of, so this is Ali Agassi. He recently graduated, and he did not just the artistry, but also the technology that underlies this. He's now at Georgia Tech, is finding plumes of contaminant buried in the earth. So the idea here is that someone spilled some bad stuff and it spreads out. These are dense non-aqueous phase liquids, chlorinated solvents, stuff from dry cleaning operations. It distributes itself through the earth, and then the groundwater comes along and washes it downstream. So as I understand it from hopefully I'm beginning to understand from working with Linda and Kurt, is that people want to remediate this. You want to get the stuff out of the earth or somehow make sure it doesn't move very far. In order to do that effectively, you need to know what's down there. And you can't just dig it all up. That kind of defeats the purpose in a sense. So we've spent some time looking at ways of trying to characterize the subsurface, characterize the distribution of these DNAPLs in the subsurface. And this project that Ali worked on was a physics-based approach. So here we were allowed to use observations of downstream contamination, so sort of what the groundwater has teased out of the earth, and cross-stream electrical resistance tomography surveys. So we get some almost X-ray tomography types of stuff downstream, and then we have these very strange electrical measurements cross-gradient. And this is a very ill-posed problem. If you were to go and try to image the contaminant structure, work through the physics and all of this, you'd come up with next to nothing. And just give up and try to move on to something easier. Ali was a really bright guy and really capable, and he came up with something that was better than nothing. So as an example, this is a little outline. So if this is the distribution of DNAPL, so this is a slice, and you can kind of see these are low saturation regions. So the brown isosurface basically is relatively small amounts of DNAPL, and then buried in here, like here, there are some pixels where we have pools. So this is just two slices. So the data that you get is we get three strings of cross gradient ERT, and then you would see down gradient, you would see this blobby image in the best of all worlds, representing sort of how much of this bad stuff the nominal groundwater flow picked up. So you almost see the shape here certainly constrains the problem, right? It's reflective of the general geometry of the structure, but in fact, if you use the right physics, which is flow and transport of multi-phase flow and transport through heterogeneous media, through the heterogeneous earth, porous media, you can do a bit more. And what the bit more that Ali came up with was this. So he was able to ignore the math, so if this is the true, it's not there, and if it's there, if you're interested, I can talk about it. I love talking about math. That's the best thing I love. It's just cool math. So if this is the sort of glove-shaped region that's truth, this is what we come up with. I mean, if this was a medical problem, I wouldn't be publishing. Linda wouldn't have hired me, and I would be gone. This is a horrible reconstruction from a medical perspective. But in terms of the limited data that we have, and in terms of the limited data and really the complicating physics, this is stellar. So it's nice to work in fields where this is considered stellar. And I don't usually talk about stellar results, but really, I don't think in working this problem, we don't know anyone that can come up with even results that are this bad. This good. So we can come up with, it's really easy to come up with worse results. It's a little more challenging to come up with results like this. And this is when I talk about, or when I hear about interdisciplinary work at Tufts, I think that this is just a wonderful interdisciplinary project, because I've spent the last 4 years learning flow and transport, which is what I love learning new physics, and this is physics that's unlike anything I was taught in undergraduate school or picked up along the way in graduate school. And at the same time, we're able to solve a problem that's kind of neat. We bring some of my tools to the table, we develop some new ones, and everybody kind of gets to learn a little something new. So this is an example. This is one of the best examples of the type of tomography work that we do. I don't know if I have two students defending, one doing breast cancer work who's done similar stuff here, and another who just defended today doing X-ray CT for luggage screening. And they've also come up with some really nice stuff, but Ali's pictures are just too beautiful to pass up. So this is one class of problem. The second class of problem is image segmentation. So back to those image segmentation. So I kind of flipped the order here. Most of you probably know can identify with image segmentation problems better than tomography, but I have a twisted perspective here. Historically, I got into this because I really love these inverse problems, these image formation problems, and I really only came to pixel processing when I came to Tufts. So the problem here, the one that led to the Ricci Prize, is this is Emilio Vasquez-Reina. He was a CS student that I was able to supervise who did this work in Connectomics. And the idea here is that you have all of this data. This is electron microscopy data of a mouse brain, and the neurobiologists at Harvard want to track each and every neuron. And this is just a teeny-weeny little portion of it. There are thousands or millions of neurons, I believe. And in fact, I was looking up a proposal we wrote For a full mouse brain, you'd have over 3 petabytes of data. So this is unlike the hydrology problem. This is a problem where we're drowning in data, and the data are really horrendous, right? So this is what's a neuron here, what's not a neuron here. You collect data slice by slice. How can you track? And the slices aren't always well aligned, and the spacing issues. So the general processing flow is that you get these slices of data, you register them, so you kind of line them up. And then we do slice-by-slice segmentation, so that on every slice we try to identify regions that are interesting, and then somehow we connect from one slice to the other, regions that look good. And this is a harder problem than I'm giving it credit for. And in the end, you get something that looks like this. So this is a visualization of all the neurons in the corresponding data stack. And I think it's generally regarded as a pretty good representation. So the problem that I'm interested in, or that's relevant here, is this slice-by-slice labeling. So if you look on these images, there are really 4 or 5 different things of interest. There are membranes, so there are cell membranes and there are myelinated cell membranes. And then there are these confusing things. Synapses have some boundaries there. These vesicles are these little circular things, and the mitochondria are these dark things. And we don't care about mitochondria, we don't care about synapses here, we don't care about vesicles, we only care about membranes because we'd like to track the membranes. We'd kind of like to find every possible tube that goes through this thing. So from an image processing perspective, the idea here is that we want to take these pixels and we want to label them. So every pixel in this image gets a label. Color, mitochondria, cell membrane, myelinated membrane, synapse, vesicle. I think those are the 5. So this is a problem in image processing. It's a problem there are a ton of ways to address it. And Emilio is sort of a side thing, actually. He had to build one, so he came up with a way of dealing with it. So the way that the way that Emilio looked at it is, look, we have people labeling these things, right? Right before we started this project, the biologists at Harvard would sit there with their grad students or their postdocs, and they'd circle all the cells, and then they track them from slice to slice, which is a horror show. But once you've done that for some of them, you have exemplars of sort of cell membranes or mitochondria or vesicles. So we can take that data and we can learn, essentially, machine learning, data mining, I don't know what you want to call it, and we can learn a classifier. So a classifier then takes, for every pixel, we take a little region, we extract some features, and then we figure out and we train a classifier that says, look, this is what everyone else thinks, this is what the humans thought, these pixels look like, well, so what should this pixel be? And you end up with going from here to here. Okay, so every pixel here is labeled by a classifier. So the classifier, you think of it as a computer program that basically looks for patterns that are identified by humans. And again, I'm doing a horrible disservice to my community by summarizing it like that, but at a high level, that's basically all that it does, right? I'll never get any funding if I said that, but it's the way that it works. So Emilio built a classifier, and this was totally tangential to his work. The reason he ended up having to build a classifier was because the people that he was working with who were supposed to build the classifier didn't come through. The work that they were doing wasn't good enough to support the type of tracking algorithms that he wanted to build. So he built this classifier, okay? And this is where Ricci comes in. So Emilio's building this classifier, and Kyung comes to me, and he says, I have these microscopy images, and they look like this. Okay, can you find the little white dots, which is the fat cells, right? So I think of things as in simplest terms. I guess they're called adipocytes, but basically it's something to do with fat. What do I know? Okay, and I'm thinking, well, let's see. Well, I can't do anything because I'm just a professor, but I have this student, Emilio, and Emilio has worked with images that look like this. Well, if I squint real hard, this and this really don't look all that different. So I'm thinking, well, okay, I'm not going to sidetrack Emilio on this, but there are a lot of cool undergrads here. In particular, Esha John, who's not here, she's now a PhD student at Columbia. She was a senior last year, and she started this the summer between her junior and senior year, was looking for an image processing project. She had worked with Professor Lasser, Ron Lasser in my department, her sophomore to junior summer, and I said, this works out, because at the very least we can take some of Emilio's ideas, throw that data in there, and see what happens. Perfect summer project. I had some extra money thanks to Linda in my discretionary account, so I could pay her, and it'd be a fun chat, a nice opportunity to get some work done. Maybe establish a relationship with Kyung, and it's always fun working with the undergrads here on research. So let's give it a shot. And that's really where it developed. So we did some work over the summer, and then Esha decided to turn this into her senior capstone design project. And then I think it was Kyung who said at the beginning of September last year, there's this Ricci Prize. You guys want to try it? They were like, sure, and I was like, sure, not realizing that I was signing up for a talk a year later. But what can you do? And it tended to work out. So it's a really nice Tufts-oriented story, right? This went from a summer research project to a capstone design project to this Ricci Prize, and I think hopefully continue collaboration with Kyung if we can both, for some reason we both ended up as chairs. I don't think either of us were chair. If we can find some time in our schedule to move things along. So this is kind of where I came in. I have no clue about fat, except that I have some. I have no clue about the biology or the chemistry. My biology and chemistry is from the 9th and 10th grade. But at least I was able to offer some services to Kyung in terms of image processing to help him say something quantitative about fat. So I think he's up next. Like I said, so this is my view of what Kyung does. He takes his microscope, he analyzes this, and he comes up with nice information about the distribution of fat. So with that, I am on time, not bad, and we'll turn it over to Kyung.
Thank you.
Sure.